convoluted tube.
In my head, you know, so that way I know that I can. And then I always have D.
So what we're going to talk about next is actually a repetition if in Bio 430 you learned about secondary active transport. You learned about secondary active transport in Bio 430. You've already heard the essence of this process. All right? So we are in the proximal convoluted tubule. And we are talking about reabsorption. So reabsorption is when we take what's been filtered and is returned to the blood, and what vessel contains the blood it's returned to. We're in the proximal convoluted tubule. What are the blood vessels that surround the proximal convoluted tubule? The peritubular capillaries, all right? So I'm going to draw a simple cuboidal cell. The filtrate's going to be over here. All right? Here's our simple cuboidal cell with the long microvilli, although I'm not drawing them in the same proportion of length. And then over here, we have the blood of the peritubular capillary. So when we talk about reabsorption, we're talking about the movement of content from filtrate to plasma. Okay? So we filtered it, and now we're reabsorbing it back into the plasma that we just filtered it from. And when we do filtration, all right, and we remove sodium ions. There's still sodium ions in the blood. We haven't filtered out every single sodium ion, all right? It's just as it flows through the glomerular capillaries, it gets some of those ions, some of those amino acids, some of the glucose will move into the filtrate. So as that content of our filtrate, we're going to have ions, and we're interested primarily in the sodium ions, all right? We're going to have amino acids, because those are small enough to move through the pores and the filtration slits. But no proteins unless we have a damaged filtration membrane. We're going to have simple sugars, monosaccharides such as glucose, because those are also small enough to move through, all right? So these are the structures that are listed over here, all right? And these first three are what we're going to focus on. Okay. Now, on the microvilli, we have various protein channels. And the process we're going to talk about involves sodium. So this is a secondary active transport. Then down at this end, we have active transport, all right? Which one of these requires ATP? Active transport. concentration, which way are our molecules moving? Down their gradient or up their gradient? Up the gradient. So from low concentration to high concentration. That requires energy. All right? Secondary active transport is occurring down the gradient. So sodium is higher outside a cell or higher inside a cell? Outside. So we're going to have a higher concentration of sodium ions here than we do inside the cell. All right? We're going to start with our active transport, because what we want to do is lower the number of sodium ions inside the cell. What's that going to do to our gradient? It's going to make it steeper. Okay, it's going to make a bigger difference between how many ions we have outside the cell and how many ions we have inside the cell. So we turn on our pump first, and that's going to pump the sodium ions from inside the cell to outside the cell. At the same time, it's going to pump potassium ions from outside the cell to inside the cell. Okay? Now the potassium ions end up leaking back out again. So we're not going to worry about them anymore. Do we call this a symport or an antiport? Antiport. Because they're going in opposite directions. All right. Now we have our secondary active transport. Sodium has a higher gradient outside the cell, so it's going to ride its gradient into the cell. Now, as an ion, it's not going to go through the plasma membrane, all right? So it needs an ion channel or some kind of support. So sodium is going to ride that gradient down its high concentration to its low concentration, but it's going to take with it another molecule that would have a harder time going into the cell. And that other molecule is glucose or amino acids. Okay? Antiport or symport? Symport. So because we've lowered the number of sodium ions inside the cell, that makes this more likely to happen because we've increased the gradient of sodium. So as sodium moves with this protein's help, the channel for sodium, that pulls with it glucose or amino acids. And that's called secondary active transport. All right? Now to get out of the cell, and it doesn't show that as on that component there, but I don't know if you recall, glucose typically moves by facilitated diffusion. All right, so it's going from high concentration to low. So it's going to leave the cell where it's in higher concentration now, and it's going to move by facilitated diffusion to get out of the cell. So the blood it's going into has lower amounts of amino acids because that got filtered into here. Still has amino acids, but lower than it originally did. And so glucose is going to go into that because it's going from a high to a lower component. All right, it leaves the filtrate via secondary active transport. And then sodium is going to leave the cell via active transport. Glucose and amino acids come into the cell via secondary active transport, and then they leave via facilitated diffusion. Okay? And they get into the blood via facilitated diffusion as well. Okay. Now, some of these other things like urea, which we're trying to get rid of, urea goes back into because it's soluble across the plasma membrane. Most of it will stay in the filtrate, all right, but some of it will leave. But it's those that I wrote on the board that I want you to be aware of, okay? Now, it's not shown up here, but water also moves across the membrane. So 80% of our sodium is going to move out of the proximal convoluted tubule by this method that I showed, taking with it, unless you're a diabetic, type 1, or type 2, taking with it glucose. Now, if you have high levels of glucose, it won't be able to take all of it because there aren't enough secondary active transports for that to occur. All right? But if you have normal amounts of glucose, all of the glucose will return to the plasma in the proximal convoluted tubule, and all of the amino acids will return, driven by concentration gradient. All right? So we don't have the effect of hormones or salt, so aldosterone does not work here on the proximal convoluted tubule. We'll talk about that Tuesday. It works at the distal convoluted tubule. And we're not going to talk about distal convoluted tubule tonight. All right? So that's essentially what I want you to recognize for the movement of sodium, glucose, and amino acids. Now for water, which we'll talk about more with the distal convoluted tubule, water moves via water channels. Water is not going to be very soluble in the plasma membrane because it's a lipid. So the water channels are known as aquaporins. So these are proteins that have channels in them that are polar in nature, so they allow the water to move through. All right, so water can move through the water channels, the aquaporins, driven by the sodium. So wherever the sodium goes, the water's going to follow because of osmosis. All right, so here's the water going to the, it's not showing the aquaporins, but they are there. Okay. A lot of texts don't talk about aquaporins until we get to the collecting ducts, but they're present in all of the parts of the kidney, collecting ducts, and all the nephrons except for one segment of the nephron, and that's what we're going to talk about next. Okay. Now, well, in 1 second. So this is reabsorption. All right. Reabsorption is filtrate back to the plasma, reabsorbing back to where it was. Any questions on this? Okay. So now secretion is in the opposite direction. So this is just a little animation showing what we just talked about. All right, so it's flipped on the side I showed you. So after transport, it's taking out sodium. That decreases the amount of sodium in the cell, and that draws in, by diffusion, the sodium and glucose, or sodium and amino acids. Okay? And then here's glucose leaving through its facilitated diffusion. All right, so secretion then is in the opposite direction. So secretion is when we have material in the plasma that we want to get rid of more than we actually filtered. So in addition to what was filtered, we're going to move more material into the filtrate. And this is showing the amino acid glutamine being broken down into ammonium ions. And we're going to talk about this more on Tuesday when we talk about buffering systems, okay? So for now, I just want you to understand the direction. So when we talk about our acid-base balance, we're going to talk about secretion of hydrogen ions, okay? And in a few segments of the collecting duct, we have secretion of bicarbonate ions. So reabsorption is going from filtrate to plasma. Secretion is plasma to filtrate, in addition to what was already filtered. Okay, so we don't necessarily secrete stuff that was never filtered. It's just in addition. So after reabsorption, all right. So if we're talking about reabsorption, after reabsorption, the fluid that is leaving the proximal convoluted tubule is going to have less sodium. It's going to have no amino acids and no glucose than it did when it was filtered. Okay. After secretion, it's going to have more because we're adding to that component. All right. So that brings us to the last concept of urine formation. I do want to talk about characteristics of urine. The last concept of numbers per volume, right? The concentration has to do with numbers per volume. So if we're concentrating the urine, we are increasing the number of particles in the urine, but either adding more particles or leaving the particles the same and removing the water. You can go either direction. So we want to concentrate the urine. When we want to remove the water, we return it to the body, either because you're dehydrated or because your blood pressure is too low. All right? So those are the major reasons why the fluid would be concentrated. Now, if we're looking at our nephron, and when I'm not going to draw the very complicated part, it's a simple nephron here, okay? So we have our filtrate. We remove amino acids and we remove glucose. And we remove 80% of our sodium and chloride, because chloride goes along with that. When we do this, all right, some of the water moves with it. And so the concentration or the osmolality of our filtrate, as it leaves the proximal convoluted tubule, is 300 milliosmoles. Now that's the same concentration of particles in the blood. All right? So the blood in our blood vessels is also 300 milliosmoles. Whether it's in the capillary, the glomerular capillaries, or whether it's in the peritubular capillaries. Even with all this moving around of particles, enough water moves or doesn't move to keep the particle concentration the same. All right? So 300 milliosmoles in our filtrate.
This is after reabsorption.
This is after reabsorption, right? Because water moves with it. So just because we're taking out particles doesn't mean it becomes more dilute. Water will move out with it. So our concentration stays the same. Okay? Now, it goes through the nephron loop, and what we're talking about here are juxtamedullary nephrons. Nephrons whose nephron loop dips down deeply into the medulla. Okay? So that's going to be occurring here. So we're going to have the medulla here. So when the filtrate enters the descending limb of the nephron loop, it's 300 milliosmoles. When it leaves, it's 100 milliosmoles, which is dilute. Okay? As it starts to go down the collecting duct, it starts out at 100 milliosmoles. As we'll see on Tuesday night, we can still lose reabsorbed sodium ions. Okay? This is under the influence of aldosterone. So on the proximal convoluted tubule, this has only to do with how much sodium we have in the flow rate. It's not regulated by sodium pumps affected by aldosterone. Here, aldosterone increases sodium ion reabsorption. Okay? ADH works on the collecting duct. Now, so obviously we removed some of our sodium ions, okay? The purpose of the nephron loop is to set up this concentration gradient. So notice the extracellular fluid around at the beginning of the medulla is 300, same as our filtrate, same as the blood. So our extracellular fluid is also 300 milliosmoles. So the blood is 300 milliosmoles, the extracellular fluid is 300 milliosmoles, the filtrate is 300 milliosmoles. As we move down, this increases get is 1,400. So 1,200 is often the number given, but the maximum we can get to is 1,400. And certain desert animals, like kangaroo rats, they can get to 4,000, because they have a longer nephron loop. So what I want to talk to you next is how do we do this? But the purpose of the nephron loop is to form this gradient. The purpose of the gradient is to be able to concentrate the filtrate. So although we'll look at the role of the collecting duct more on Tuesday, let's say, so this filtrate comes here at 100 milliosmoles. If you drank 3 gallons of water since 6 o'clock last night, do you need any more water to be kept in your body? After drinking 3 gallons since 6 o'clock last night? Probably not, okay? So the urine that is released is going to be a very light, pale yellow color, and when it leaves, it's going to be 100 milliosmoles. We don't get it more dilute than that. What if you haven't drunk any water since yesterday morning? Your body's going to be dehydrated and it's going to need as much water as possible. So ADH's role is to, and I don't have cells here, but it causes water pores to be inserted into the membranes of the simple columnar cells, a few or a lot, depending on how dehydrated you are. Now, as the 100 milliosmol flows into a section of the medulla that is 300, and we have aquaporins, what's going to happen to the water? It's going to move from a less salty area to a more salty area, right? Remember sugar on top of strawberries is going to draw the juice of the strawberries out? So if we have an osmotic concentration of 300 here, water is going to be drawn out to the higher concentration of salts and a little bit of urea. All right, so now this becomes 300. But as it flows farther down, it gets to an area of the intracellular fluid where it's 500. 500 is more concentrated than 300. So if there's water porins present, even more water gets pulled out, and our filtrate becomes concentrated to 500. And you can see where this is going. So as it travels down to the tip of the papilla, it's getting to more and more concentrated extracellular fluid. And if the water channels are present, water moves out and the filtrate becomes more concentrated. Okay? So the dynamics of this are that the function of the nephron loops is to set up this gradient. However, they do not, the nephron loops, and this is important, the nephron loop does not function to concentrate the filtrate. Why? Because it's actually diluted by the time it leaves. The filtrate, when it leaves the nephron loop, is only 100. It's diluted, it's not concentrated. Where it becomes concentrated is in the collecting duct under the influence of ADH to add the aquaporins because of the gradient set up by the nephron loop. So the reason it's diluted is because we took ions, we took sodium and chloride ions out to make this more concentrated. Therefore, the filtrate is more diluted. Okay? It's easier to add or remove aquaporins than to change the gradient, depending on how much water you drink from day to day.
The 300 to 1300 is outside of the loop?
Yes, we'll see
Okay.
what changes inside, because this will change too as we pull ions out. But I wanted to show you the purpose first before we look at that process.
And it's setting up the outside.
Exactly. So the entire medulla
Okay.
of all of our pyramids is going to have this gradient. Okay? Yes?
Just making sure, so the function of the nephron loop is not to concentrate the filtrate.
Right. It's to set up the gradient of osmosis so that if needed to be concentrated, that occurs in the collecting duct.
And the concentration gradients.
So the nephron loop forms the concentration gradient, but it doesn't itself. The filtrate is not concentrated when it leaves. The filtration, the concentration occurs if needed in the collecting duct of the medulla. Collecting ducts and the cortex can't concentrate because they're completely surrounded by 300 milliosmoles. Okay? All right. So, now let's look at that process. is known as countercurrent multiplication. We've already talked about countercurrent flow, right? We talked about that with the heat exchange in the testes with the pampiniform plexus. And the hot arterial blood was the countercurrent flow to the colder venous blood coming out of the testes. And we use that for heat exchange. The reason we call this countercurrent multiplication is because the fluid flow is connected here at the bottom. So in our venous and arterial flow, we had two separate vessels going past each other. Here, what was flowing down now is countercurrent because it's flowing back up. And that magnifies the ability to exchange, okay? Secondly, we have pumps, sodium, potassium pumps, all right, that are gonna move sodium ions out of the filtrate. They have a limited maximum. They can only create a gradient difference of 200 milliosmoles. So there can only be a difference of 200 milliosmoles between what's in the filtrate and what's outside the filtrate, okay? However, 1400 is very different from 300, right? So the reason we couldn't do that with just pumps all alone on our, if we didn't have a countercurrent multiplier. The maximum difference we could have was 200. We could go from 350 to 550, okay? So we're gonna go through this slowly because it kind of shows you how the pumps work. So here's the, actually again, I did it backwards because this is now the descending limb and this is the ascending limb, okay? So we're gonna start out without the pumps working at all. We're gonna fill the descending and ascending limbs of the nephron loop with 300 milliosmol filtrate, okay? So we don't have any pumps working right now. We don't have the countercurrent flow working. Then we turn on our pumps, okay? And so remember, this is the ascending limb. And it helps for you to remember that if you remember that when we leave the ascending limb, we are at a dilute. We are at 100, all right? So we pump out sodium, chloride follows passively, and we go from the ascending limb into the extracellular fluid. And it shows it in the middle, but it would be all around, okay? So, first rule, ascending limb has the pumps, okay? Second rule, the ascending limb is impermeable to water. Water cannot leave. This is the one place in the entire kidney where there are no aquaporins. We have aquaporins in the proximal convoluted tubule. We have aquaporins in the descending limb. We have no aquaporins in the ascending. Okay. What would happen if we had aquaporins there? The water would follow, and there wouldn't be any purpose in using the pump, okay? So, we've moved, we now have a 200 milliosmol gradient present. Okay? But before we had 300 on both sides, we take out 100, we add 100. That's as much as the pumps can maintain the difference without the sodium leaking back in, okay? So, because this is higher than this, remember this is the descending limb, the pumps are in the ascending limb, water can't follow, so it becomes more dilute as we move out the sodium. However, in the descending limb, water is permeable. And so the water here is going to be moving by osmosis to the higher particle concentration. What's that going to do to the concentration here if we move out water? It's going to go up. Okay? So now we pump out more, and water follows, and pump out more, and water follows, and we pump out more, and water follows. But all we did was got to a maximum of 400. We're not nearly to 1200. Okay? All right. So now, because this is countercurrent multiplier, we're going to take this 400 and start to allow it to go back up here. Notice how what's leaving is more dilute than what was coming in. That's how we get to 100. All right? So as the fluid leaves the ascending limb, it's dilute, and that's why, because we're moving out ions. Okay, so we're now going to let this move. We're going to move over here. We're now we have 400 on this side. Okay? So now we pump out more. And 500 is higher, still higher than 400. So water moves out, and this becomes 500. And we have a gradient from 350 to 500. Okay? So now we move the 500 over here. Again, we can maintain a 200 milliosmol difference. Now we have an even more fine gradient. So now we move the 600 over. Alright, and by doing all of that, so on, we get our gradient of 300 to 1200 or 1400. Notice how this is only 200 milliosmoles higher than its counter level in the ascending limb. That's the maximum difference we can maintain with our active transport pump. Whereas on the descending limb, it's the same as the surrounding fluid because it's been equilibrated by the osmosis of what's in the extracellular fluid. That's the multiplier effect by taking what's in this limb and moving it out here. So the ascending limb pumps out the sodium. That's how it gets diluted. It's impermeable to water, or the water would just flow and it'd stay the same osmotic concentration. The descending limb is permeable to water, and that water is pulled out to match the gradient, and so it becomes more concentrated. There's a question here that you should ask. Do you know what that question is? What did I do over here? Kept flowing out water, right? Water should be diluting the sodium that I'm putting out here, but it didn't. I ended up with a gradient of sodium, and urea's in there too. Why did that water end up diluting this and messing up my gradient completely? What are the vessels that we have in that region? The vasa recta. The vasa recta picks up that water, as well as any water pulled out here as we concentrate the filtrate and carries it away. And helps to maintain that gradient rather than getting diluted by all the water coming out. Okay? Let's go through it one more time.
What do we, I understand the concept, but what do you want us to know? Do you want us to let the numbers on this?
So if I were to give you a loop like this, okay, and I were to tell you, I would say, well, first of all, I want you to know that the ascending limb has the pumps. The ascending limb is impermeable to water, and the descending limb is permeable to water, okay? Then in addition to that, if I were to tell you that the ascending limb was 200, and I asked what was the osmolality of the extracellular fluid, I would want you to be able to say it's 200 milliosmoles higher.
Okay.
And it's going to be the same as whatever is in the descending limb too. So by the time we're done with all of this, the extracellular fluid is going to match the descending because it's equilibrated, and it's going to be 200 milliosmoles higher than what's in the ascending because our maximum difference is 200. Okay? So that's what we use the juxtamedullary. That's the purpose of the juxtamedullary nephrons, is to send their nephron loop deep down into the medulla to, through countercurrent multiplication, form this gradient. So that if and when needed, water channels in the collecting duct can allow that osmotic gradient to remove water and concentrate the filtrate. If you've drunk plenty of water, then you do not insert. The role of the antidiuretic hormone is to instigate the insertion of those water channels. If those water channels aren't there, the filtrate stays diluted. Okay? Now it might be 300 instead of 1200, in other words, only a few water channels were inserted. Alright? If no water channels are inserted at all, the urine is at 100 milliosmoles. That's as dilute as it can get. Okay? Alrighty? Okay, so before the lab is very basic.